Hello, A&P1 students, and welcome to the Chapter 14 presentation on the biggest mass of neurons in our body, the brain and cranial nerves. The brain is a concentrated aggregation of neurons that are arranged together in such a dense packing that we cannot follow individual nerve pathways. We have to rely on the sum total of those electrical discharges in order to conceive of how the brain works. So the idea in the brain is there are going to be regions where there are emergent properties from the collection of neurons, not relevant to think just of individual nerve synapses or nerve-to-nerve connections anymore. The brain functions include storage and processing areas, relays, and connections, but always the same kind of organization: incoming sensory information, information processing in the gray matter of various areas. And here you have different information processing areas combining to produce basically a motor response that gets sent out through the peripheral nervous system to our effectors. There are going to be 6 regions in an early figure, and I want you to know each region, and I want you to know a general description of the large functions that occur there. The brain, like the spinal cord, is separated from its surrounding tissue by the meninges, the dura mater, arachnoid mater, and P mater that we introduced before. But it's such a dense collection that it's no longer okay to just have a trickle of cerebrospinal fluid. We're going to have cavities and channels for the cerebrospinal fluid to flow through. They're called the ventricles, and they're a real part of the brain's structure. The regional landmarks include anatomical clues, little lobes, little splits, little cavities, and there's a specialized anatomical terminology for those features in the brain. We're going to talk about the blood-brain barrier. How does blood get resources into the brain, and what regulates that? Attached to the brainstem, there are 12 pairs of nerves that are indicated by that capital N, followed by a Roman numeral 1 through 12. And the cranial nerves, although they're within the skull, they're attached to the brain stem. They're considered part of the peripheral nervous system. The basal nuclei are an example of a particular aggregation of parts of the brain that have been correlated with specific functions. So let's delve into this. The organizing principle is related to the structure of the neuron, mixture. So brain mapping shows this complex connection and interconnection of branching cells that basically reach out in all directions and form synapses with surrounding cells. Neural structure and function changes throughout life. So the brain not only grows physically, but it learns, it adapts, it changes throughout our life. Neurons are neurons, so they interconnect freely, and it's that growth that generally involves the development of our brain and brain capacities. Sometimes you will see a nerve and separate that nerve anatomically from its surroundings, and it is generally physically and chemically separated from surrounding tissues, from the plasma side of the body. Often a nerve will be primarily sensory or primarily motor, but they all often involve a mixture. So let's take a look at this brain outline. This is the leading figure from your text, and it introduces the brain and its 6 regions very clearly. Once I get my pointer on, I can basically look at this entire brain and the brain stem, showing how it sticks out at the bottom. And this is embedded. If you were looking at this from the front, the line of this brain stem would be right on the midline of the body. So these 6 regions begin to be called the brain and brainstem when you cross this border. Do you see the change in shading right there? Below this is the spinal cord because you're below the foramen magnum. Once you travel up the spinal cord past the foramen magnum, you have the first lobe, the lower lobe of the brainstem called the medulla oblongata. Here we have, of course, relay information. Anything that gets back and forth between the body and the brain has to go through the medulla. So there are relay information up to the thalamus, which is going to contact the cerebrum, back to the pons, up to the pons, which is going to connect to the cerebellum and to all other parts of the brainstem. We also find within the brainstem certain autonomic centers for the very basics of mammalian metabolism. So the basic control and regulation of autonomic functions like the heart and blood flow, the respiratory system, the digestive system for mammals are located here in the brainstem. Now, here in the medulla. You can also see these wiry or fibrous kinds of connections. These are the cranial nerves, and we'll save their identification for later. Here, this bump, which is prominent in the tissue and shaded blue, is the pons. The pons is another relay, but it's also a fork in the road. From the medulla, you can go up to the diencephalon and out to the cerebrum up here, or you can turn the corner and go back to the cerebellum. The cerebellum is a region for motor programs, so the pons is its primary connection to the rest of the nervous system. So these relays and this Y, this fork in the road, is important for the pons. We also see subconscious, somatic, and visceral motor centers in the pons. So it's related to the motor programs in the cerebellum. More about that in a moment. The midbrain, right here, this green region, has centers for the processing of visual data and auditory data, the generation of somatic motor reflexes and responses, and also maintenance of consciousness. I never thought much about asleep or awake, but it turns out those two states of awareness are something that the brain actually does. The brain puts you to sleep and the brain maintains your wakefulness. Up at the top, this lobe at the top is the diencephalon, and it looks like a single lobe from the side because the left side obscures the right side that's over on the other side. Here we have the hypothalamus, which is the floor of the diencephalon. Basically, all this tissue is called thalamus tissue. The thalamus is a big relay center because whatever travels up the brainstem like this and gets up here to the diencephalon is then going to be routed to an appropriate place in the cerebrum. So big for relay centers and for dividing signals to feed single sensory inputs into multiple regions of the cerebrum. But the hypothalamus is also very important. The hypothalamus, which means below the thalamus, that's just basically the floor of the diencephalon. And it is the center that links brain function to endocrine function and hormone. That's typically due to the hypothalamus and the pituitary gland. And we'll get more into that in a bit. So this is that central core. You can see it kind of shaded in here. The connection of the pons to the top of the cerebellum. The other two lobes, the cerebellum, very densely folded in the second biggest lobe of the brain. It coordinates motor programs. That's what I want you to remember about the cerebellum. This is where we store our skill memories. So as we learn to hold up our head, to direct it towards certain sounds, for example. As we learn to hold our body erect and eventually stand and walk. Those are all being programmed here in the folds of the surface of the cerebellum. This is where our motor performance actually exists. So if you took piano lessons and were accomplished, whatever you learned was programmed in here. If you were an athlete and learned the pole vault, your basic understanding of the feeling of doing a pole vault was programmed here. That leaves us with the sixth lobe, this outer cerebrum. And one of the things you notice about the bigger lobes, the cerebellum and the cerebrum house, how they look so folded. These folds develop through life. And as before, what did it mean on the cellular level when we saw folded membranes or growths like little fingers or bunches of grapes? Those meant increased surface area. And in fact, that's exactly what they mean on the organ level here. These folds increase the surface area, and the surface area is where the gray matter is. We do have a couple of terms, these ridges that seem to squiggle back and forth. That's called a gyrus, and the plural are gyri. The gyrus represents the ridge. The fold between them is called a sulcus. The plural is here, sulci. So this ridge and fold arrangement is actually one that's fairly, not exactly alike from person to person, but there are reliable deep folds called fissures where the fold goes way down in and separates the brain into lobes that roughly correspond to the brain, to the skull bones that are opposite them. So the frontal lobe is right below the frontal bone. The parietal lobe, the occipital lobe, and the temporal lobe are generally divided by very deep fissures. When we look at the development of the brain, we have always the same initial thought. We start out as a single squishy cell. There's no sign of a brain. There's just one cell. So what happens as that cell divides and begins developing tissue? When we look at the development of the brain, here's the primary brain early in development at just a simple tube. By the way, that is an eye spot. That will develop into optic tract. But there are three basic lobes, 1, 2, 3, called proencephalon, mesencephalon, and rhombencephalon, which continue to grow by cell division and cell differentiation. So at 6 weeks, we definitely see a folding and a development of these regions, and we can basically identify these lobes and regions with what will be this part of the brain at birth. So the telencephalon becomes the cerebrum, and the diencephalon becomes the diencephalon. The mesencephalon becomes the midbrain of the brain stem, the metencephalon region, the cerebellum and pons together, and the myelencephalon, the medulla oblongata. So this is basically just an embryonic translation table showing you how with more cells and more elaboration, we develop these recognizable birth regions. We also recognize the formation of certain cavities and certain channels to connect them. So in the cerebrum, we see the development of lateral ventricle, first and second, left and right, because there are two lobes of the cerebrum. The third ventricle develops right by the diencephalon at the top, above the brainstem. The cerebral aqueduct through the midbrain connects to the fourth ventricle, which is spanning this cerebellum and medulla oblongata region. This will produce a continuous channel for the flow of the cerebrospinal fluid. This shows in highlight form the presence of those ventricles and their connections. So from the side, you can see these blue channels are looking like very large circular objects with a horn in the back to basically reach the major channel to be within a certain distance from the surface of the cerebrum. This third ventricle looks very large indeed, and in fact connects to the left and right laterals, which would be the 1 and 2. And then the cerebral aqueduct connects down to this fourth ventricle. But turn the brain 90 degrees and look at it head on. You'll notice that those two cerebral hemispheres have the lateral ventricles within. They look kind of like ram's horns, and they swoop around in that circular pattern, traversing all of the lobes from frontal to parietal to occipital and down to temporal. You notice how they flare outward from the midline, and behind them you can kind of not see the spike that goes all the way back to the occipital. This third ventricle, which looked so large from the side, now looks narrow because it's mounted right on the midline. But it connects this upper region across down to the top of the brainstem. Here's the cerebral aqueduct from the midline again, and then widening into the fourth ventricle and changing into, of all things, the central canal. The brainstem central canal is continuous with the central canal of the spinal cord that we've seen already. The brain is separated and protected from the rest of the body. That separation is physical, and the large part of that is accomplished by the dura mater, but it's also biochemical. And here is where the wrapping of capillaries that penetrate the brain by those astrocytes and the regulation by satellite cells becomes very important. What a layer of protection. This is the most protected part of the body, the skull, cranial bone, which is strong and hard at maturity, followed by fluid-filled layers of meninges and what are called dural folds. Now, when you fold the dura mater, you create a system of channels and also a system of tough membranous material that will separate major lobes. We'll look at those dural folds in just a moment. Inside the cerebrospinal fluid is the chemical environment of those neurons and all that other tissue. And in addition, because it's a hydraulic system, that fluid provides a cushioning against shock. Neural tissue is very subject to shock because of the synapse. Jar it too much and change that synapse. Open it up or close it down too much. And the brain loses some of its connections. We talk about this isolation as the blood-brain barrier and acknowledge that the conditions of the body that are so universal elsewhere are not maintained in the brain. It's a special environment. Here we see the protection. Here's that bone of the cranium. Here is the dura mater, outer layer and inner layer, and the dura mater has a dural sinus, which contains cerebrospinal fluid. Now I'm going to go to the other side. See this cerebral cortex? That's the brain tissue, and this is the pia mater, and then this spacer is the arachnoid mater. This is all permeable to cerebrospinal fluid and wet and cushioned. When you look at the brain as it sits inside the skull, you'll notice the region. When we looked at the skull, we saw right up here the base of the frontal bone, then right about this level a step down the base of the sphenoid, and then down here the very lowest part of the occipital lobe. So here and the brain fits exactly within that space. Take the neural tissue out. So the cerebrum and cerebellum are removed here. The brain stem and diencephalon are nowhere to be seen, and you see the dural folds. Now what that means is that when you take the dura mater and spread it over the top, and you get right here to the crown, there's a longitudinal fissure running front to back, right across the crown. That dural fold actually basically turns the corner and folds down into that fissure, and from both sides. So it wraps it and separates the lobes of the brain. We call these three major partitions the falx cerebri. This separates left from right. You notice not only do you have this double dural dura mater fold, but there are channels within it to help conduct the cerebrospinal fluid. We have another one called the tentorium cerebelli. It's separating the lower extremity of the cerebrum from the chamber that holds the cerebellum below. And finally, this falx cerebelli is the dural fold that encases the back, the very occipital lobe portion of the cerebellum. So dural folds, structural and chemical separation are maintained in the brain. Now, cerebrospinal fluid's been mentioned several times, but we've never said where it comes from. And it comes from a tissue called choroid plexus. For all intents and purposes, it looks like a kind of a fuzzy, ripply ribbon. It's shown here in red. And what you're actually seeing here, we've cut along the midline and taken the whole left side off. We are looking at the folds from that longitudinal fissure. This is a part that's cut, and we'll talk about the corpus callosum in a minute. But right here, we're actually looking over into the lateral ventricle on the right side, and that little squiggly ribbon is a tissue called choroid plexus. Now, that's in the right lateral ventricle. The one in the left lateral ventricle has been removed by the dissection. So two stripes of choroid plexus in the lateral ventricles. On the roof of the third ventricle, you can see a third choroid plexus strip. And down here in the fourth ventricle, a fourth one. So these glandular tissues are producing cerebrospinal fluid at a reasonably high rate. So this is something where you're pouring the cerebrospinal fluid into the ventricles, and the pressure that comes from the production is basically pushing it around through all these channels. You can see the arrows taking it down through the central canal, but also circulating through these openings around the various lobes of the brain in a complete circulation pattern. Around the end, down and back up, circulating around until eventually it climbs this space here between the cerebrum and the dura mater. Now in the top, there are little pressure valves. So here's the cerebral cortex, and here comes the material flowing through the space between the pia mater and the dura mater to this particular little pressure point called an arachnoid tubercle. This arachnoid granulation is porous and allows that pressurized cerebrospinal fluid to flow out into the dural sinus. Now there it joins a system of fluids that is cushioning, but also is connected to some porous capillaries so that the excess cerebrospinal fluid is pushed out the other end of this circulation pattern into the blood, where it is dealt with as waste. The isolation of the central nervous system is going to depend on transport across barriers that is selective and directional. The cells of the nervous system actually select. They basically divide the materials into two piles. And astrocytes around capillaries are regulating most of the molecular exchange up in the brain, the central nervous system. The blood cerebrospinal fluid barrier is also reinforced along the channels by special ependymal cells that surround the choroid plexus and regulate exchange. Only in certain regions do we relax this barrier. The hypothalamus, pituitary, and pineal glands are all glands that produce hormones. When you dump a hormone, you want it absorbed and circulating around as fast as possible. So here we find special, not just capillaries that are exposed, but special capillaries that are exposed so that these hormones can be absorbed and circulated rapidly. Let's go through this 6-region division carefully and try to reinforce some of the functions. The medulla oblongata, relays and vital centers. The autonomic nervous system is especially important here. A structure called the reticular formation is an identifiable strand of tissue that starts in the medulla oblongata, goes clear across the pons and midbrain. These are relay centers for things like cardiovascular, digestive, and respiratory function. The reticular formation produces reflexes where sensory information are converted from the cranial nerves and the cortex of the cerebrum and the brainstem. Cranial nerves that hook to the medulla oblongata are 12, 11, 10, 9, and 8, and you'll always see them as little strands sticking off of the MO, and basically always pairwise. The idea with the medulla oblongata is connect. the spinal cord below, and all the rest of the brainstem and brain above. The pons is a recognizable bulge between the medulla oblongata and the midbrain above. And the pons is the connection point for the cerebellum and the midbrain above. So it's a very significant corner. If you've got motor sensory information that affects motor performance, it's going to turn the corner there and go to the cerebellum, as well as perhaps going up to the cerebrum. Cranial nerves 5 through 8 hook on the lateral and aspects of the pons. Respiration is controlled here, and basically the pons links the cerebellum to all other regions. So here we have more relays up and down, but across now. When we look at these figures like this, there's a lot of detail there, but the main thing I want you to see are the basic areas. Here's what we looked at first, the lateral view, and I said this diencephalon looks like a single lobe. But when we turn it, looking at it from the front or from the back, we'll notice, no, wait a minute, those are two lobes that basically flare out left and right, in this case, left and right, to form a core of the cerebrum. So looking at it from behind, the lateral fissure is going to come right down here, and then the cerebrum is going to go up and wrap around that diencephalon from, in this case, from medial to lateral. And then when you look at it from the side, the cerebrum is basically wrapping around it all the way around from front, across the top, to back, and then down below. So this really very much, the thalamus and the diencephalon very much do form the center, the relay center for the cerebrum, left and right. We can notice a couple of other interesting features. So this is a very good view showing all of the connections for the different cranial nerves. And here is their numbering, all except N1, which we'll get to in just a moment. You can see some are major, like these are the optic nerves going back toward the occipital lobe, and these cut ends are the ones that are going forward up into the eye. But you can see sometimes they just peek out from underneath the pons. At other times they are distinct groups coming out from the brainstem. The last thing I want to point out on this figure, well, no, I want to do two more things. Notice these different colors and all the lobes. These are named parts of the brain, and you'll notice the odd kind of terminology like colliculi and this is specialized brain anatomy. We're not going to really focus on it much. Just know that the brain is well examined and very intricately named. The other thing you can see here is the choroid plexus. So looking at the back, the lateral ventricle would be above and all around this diencephalon. You can see the choroid plexus left and right draped over the back and kind of around the forward part of lateral ventricles left and right. Down here at the fourth ventricle, you're going to see another stripe. And finally, this choroid plexus, the third ventricle is sort of out of sight. It's concealed in this particular dissection. But down here from the back, you've opened up the fourth ventricle, and you can see the final pairs of choroid plexus. All of those are producing cerebrospinal fluid for circulation around those cavities and channels in the central nervous system. The medulla oblongata and pons are shown here with relay centers, and this color coding that's up here makes it appear very simple. The lateral white columns, the nucleus cuneatus, the olivary nucleus, each of these have respective functions, but it is very complex and kind of beyond, I believe, beyond the idea of a basic introduction to the brain. So I'm not going to require this specialized anatomy to be learned. But look here, here's a nice drawing of the pons. And the channel coming down, here's the fourth ventricle and the connection of the cerebellum in the back, showing you that this is the only connection region for cerebellum and motor program activity. The cerebellum, motor programs, that's coordinated, learned, and reflexive pattern of subconscious muscular activity. We learn by concentrating on something. Once we learn, we pattern that learning, sort of like a subroutine. We can call it any time. I know I can balance on 2 feet. I don't have to think about it and decide. It becomes almost a reflexive activity to stand on my feet and maintain my balance. I do that unconsciously by adjusting the postural muscles all up and down my skeleton, especially in aligning my pelvis and spine, which is kind of the axis of the body. The motor programs are constantly reading sensory information. The proprioceptors are constantly sending, using stretch receptors throughout the body in joints and muscles to send a constant flow about the position, the relative movement, and some balance information about the various parts of the body. And constantly the cerebellum is reading those and adjusting those postural muscles to keep the spine, the skeleton, as the center of the weight-bearing structure of the body. Now, this is an amazing figure to me. It does very well show the anterior and posterior lobe of the cerebellum. But the thing that struck me the most when I looked at it the first time is how much a fresh cerebellum actually looks like hamburger. That's what it looks like to me, as a package of hamburger. But it does set up a sectioning of this. You can see this midline section shows the cut surfaces that clearly show how the folds on the surface penetrate into the cerebellum. There typically is, in a big lobe, a white bush-like structure of white matter that basically reaches out into every region of the cerebellum. Notice that there are minor folds right here. Those two only go very shallow, whereas these are major folds and separate very large sections of the cerebellum one from the other. This is an area of very intense interconnection. That means that neurons form unbelievable numbers of connections. These are the cells of Purkinje that I've mentioned several times. And you can see that it looks very much like a mass of roots that are intertangled. These dendrites of the cell bodies are forming synapses with these other tangled dendrites and cell bodies, producing a level of integration and of, a good term that I like, skill memory. So your motor programs that become automatic, that become coordinated. The first time you swing a tennis racket, you may be clumsy, but quickly you develop the coordination of how to swing, how to shift your weight, how to coordinate your eye, and you're watching the ball with the swing of the racket, and it becomes a skill. That's all programmed here in the cerebellum. The midbrain of the pons is a short region and rather nondescript, but lots and lots of gray matter. Many inputs are coming to the midbrain that include communication from the medulla oblongata below and the peripheral nervous system. A flow from the cerebellum and pons as well are reaching the midbrain, and the gray matter there is distributing it, forming new synapses, and setting up its communication up through the diencephalon. Integration of these inputs and many innate reflexes. And here, these are things you may not have think of, like blinking the eye, something that we don't pay any attention to, but is important to the maintenance of our vision. The contraction of the iris when the light gets bright, and its opening when the light becomes dim. Head jerk response. If something flies towards your face, you typically jerk your head away to protect. We also see cranial nerves 3 and 4 attached to the midbrain. So here we see this kind of nondescript area. There are named regions. Here is the top of that reticular formation we spoke of when we looked at the medulla oblongata. But one thing that is interesting about these figures, as beautifully as they're drawn, when you actually look at the material, when you actually look at the tissue itself, it's in this preserved form, it's a kind of a series of white and gray grading into kind of a tan and light brown. You can see the folds of the cerebellum. You can see the cerebral aqueduct that will connect with the central canal, but basically it's a fairly nondescript, smooth tissue that presents itself. The diencephalon. The diencephalon is two lobes that sit and flare off the top of the midbrain. We used to think of the diencephalon as the top of the midbrain. We now know that it develops from tissue that's common with the cerebrum itself. This is where we're going to receive all this sensory information from below, from motor signals as well, and we're going to integrate sensory and motor. Thalamus is the name of the tissue, and the roof is called the epithalamus. That means the top. The main body is called the thalamus, and the floor is called the hypothalamus. We're going to see here the pineal gland as well, which is a source of a hormone called melatonin, something that many of you probably use as a sleep aid. Its sleep cycles and falling to sleep and waking up are strongly affected by the pineal gland. There is a left and right thalamus, which corresponds to the left and right cerebrum, and roughly there's a map of relays in the way that those neurons and labeled lines radiate out of the thalamus and connect into the cerebrum. The floor, the hypothalamus, is this important function, coordinating the nervous system's action with the endocrine system. To give a simple model, we're going to talk later about sympathetic nerve stimulation, the activation of muscles, lungs, and heart for maximum physical output. That's the type of thing that usually comes from seeing, hearing, sensing a threat somehow, which alarms you. And it is the activation of the hypothalamus, which signals the release of epinephrine and norepinephrine from the adrenal gland, that keys that overall body reaction for maximum activation. We'll get to that in a bit. So here we are looking at the relative position from the lateral aspect of the diencephalon, in this case on the left side, and you can kind of see the rough anterior group would be sending labeled lines up into this anterior part of the brain. Notice also that we've colored the lobes of the brain differently, and for the frontal lobe and parietal lobe, this particular gyrus on each side of this very deep sulcus have been colored with more intense color, indicating a primary and secondary function on each side. Let's take a look inside the brain. Here's a brain that's been cut along the midline, a mid-sagittal cut. And you can see inside that fold there are blood vessels, and these are arteries, so there's no need to coat them. But within the tissue, capillaries are completely embedded by astrocytes. These folds continue right down. So this even though it's within the fold, this is an outer surface of the brain. This very smooth surface, called the corpus callosum, is a series of white traces, white matter, that are running left to right across the brain, from joining the left and right side, and connecting those two at its very base. So this is the base of that fissure. Here we see the fornix region and the opening to the right lateral ventricle. You can see the choroid plexus sitting there. So this is smooth and white on the corpus callosum because it has all been severed by a scalpel. Here, right at the base of this connection between the hypothalamus, basically the thalamus is tucked up here out of sight in the right lateral ventricle, and hanging, this region is so nondescript, it's basically just the floor of the thalamus, is the hypothalamus region. And right here, we will see the region of the infundibulum. This is the stalk that hangs the pituitary. The pituitary has been removed or lost in this dissection. Up here, you see the relative positions of the hypothalamus and the suspending infundibulum and this little globular pituitary gland. Now, as unimpressive as that looks, the hypothalamus is the link to the brain. This is the master gland of the body. It produces around a dozen hormones, and each of the hormones controls another gland, whether it be the thyroid or the adrenal or the thymus, these or the melanocytes. This particular is the master gland of the body. Within these regions, we see considerable integration, what we would call a functional grouping, that cross different lobes in different regions of the cerebrum and diencephalon. And this relates to a number of diverse functions. Now, it's hard to say this in a simple way. It's not simple because it is integrated in such a complex way. The limbic system is one of these functional groupings, and you're going to hear more and more about it because it has a complex anatomy, but it's involved with emotional states. So the limbic system and its coordinated action across the lobes is going to link that cerebral conscious intellect, what we know and remember, what we think, with the subconscious and autonomic functions of the brain stem. We also know the limbic system in good health is required for good memory storage and retrieval. So the idea with the limbic system, the reason it's so much important to our health, is that it combining the intellect with the emotion, we're really talking about the how, the intellect, with the will we, the motivation. And you have to put those together to actually do anything. And so it kind of shows two different aspects of neuronal function up in the brain. This is what I mean by a complex anatomy. The limbic system is illustrated by colored pieces here. You can see it's three-dimensional, has this kind of horn shape curling around the corpus callosum on left and right sides. Is this green region called the cingulate gyrus. And this gyrus starts here in the frontal lobe, right above the pituitary and hypothalamic region, goes anteriorly and superiorly, a single gyrus, around through the frontal lobe, right across into the parietal lobe, all back here, right across into the occipital lobe, and then swooping down to the side, you can kind of see that here, and connecting to other regions. These regions then spanning down into the temporal lobe. So a functional grouping that crosses and connects all parts of the cerebrum is what the limbic system is. Within it, we see the hippocampus, the parohippocampal gyrus, the mammillary body and other well-defined structures, the amygdaloid body. Each of these is now being examined for more specific information on how it affects the integration of intellect and emotional states. The cerebrum, our largest lobe. This is the center of processing, of intellect, and of higher order functions. Now, that doesn't mean it's not involved with things like motor movement. We map the sensory input and motor output to our body, to our skeletal muscles, to the cerebrum in a specific way. We'll show that in just a second. The cortex is the wrinkled surface with gyrus and sulcus anatomy that increases its surface area. When a baby is born, its brain is smaller, but it's also just kind of wavy. As your skull grows and your brain grows, it not only gets much more much greater in size, but it wrinkles up to produce that increased cortex, the cerebral cortex. We find that to be a layer of gray matter right next to the surface, and then basically connected from below by white matter strands that provide connections to other places of the brain, and then feed those signals out of the brain and down to the peripheral nervous system. So white matters for relays cross the brain at all times, and from all positions to all other positions. So left to right, front to back, there are white relays that provide an integration of all functions. When we look at the brain in its preserved form, it looks like this, kind of ordinary and kind of brown, wrinkled, and with all of those lobes that we mentioned. When we color code it, we see a couple of features. We see this central sulcus dividing the frontal from the parietal lobe, and the frontal lobe, shown here in pink, is also divided from the temporal lobe by this lateral sulcus. So this is a very deep channel. This is a very deep channel. This lateral sulcus, in fact, if you use retractors here and pull it apart, down it's kind of a T-shaped hole with the sides of the hole and the back wall wrinkled. This is essentially like an outer surface of the brain. The occipital lobe in the back is kind of isolated, but does have its sulcus divisions from the surrounding lobes. When we look for association areas, meaning can we localize any specific region of the brain with a specific function, we find a very general and very consistent division of those functions. So for the frontal lobe, we have the prefrontal cortex, which is apparently a region of like short-term memory. When you read, when you learn something for the first time, this is where your short-term memory is. It is short-term. It'll be replaced by whatever flows in next. It has to be passed throughout the brain in order to become a long-term memory. Here we have on the frontal lobe a motor association region, meaning that this is the secondary region and this is the primary region. So we process the motor commands and responses here, send them to the primary region for communication out to the body. Right across this central sulcus, we have a similar organization on the parietal lobe for the sensory cortex. So here's where we receive the sensations, and this is where we begin that processing, the secondary processing region. Then those signals are networked throughout the brain to generate those motor responses, which are collected here, communicated here, and then sent back out to the body. We do see a couple of logical connections. The special senses, for example, this auditory association complex is on the frontal lobe, right in the region where the internal auditory meatus opens into the skull region. So that kind of makes sense that hearing would be here. But what about this? Here's the occipital lobe. Here's where our vision is processed, is way, way back here on the opposite side of the skull from the actual eye and orbital cavity. The smell and taste, here is olfactory, which is right on the temporal lobe, on this basically the side wall of this lateral sulcus. This is where we smell. And gustatory is on this back wall of the frontal lobe. This is where we taste. We have made tremendous efforts to divide and recognize different features of the brain. In some cases, this has been successful. Here's a general interpretive area. This is specific regions on the frontal lobe. For the frontal eye field, the speech center have been localized to the frontal lobe. This is the beginning of a drawing that was developed by a physiologist named Brodmann. by looking at the histology of different tissues, and you can see the different numbers and regions. His hope was that the functions of the regions of the brain would be distinguished by their different histology. In some cases, this did yield some valuable correlations like 18 and 17 and their association with visual processing. However, in other cases, there was no apparent simple correlation with a recognizable function in the body, a tribute to the integration of neurons in the brain. So when we look at the cerebral hemisphere from the front, we do notice a couple of features. One that is good to relate to the visual fields. The visual field, left red and right blue in front of us, is basically brought through the lens of the eye, where it is inverted top to bottom and left to right. So the left side up would be the right side down on the image on the retina. This would be happening in both eyes. These are creating images back here on the retinal surface, which are fed through the optic nerve back to this chiasma. Now, a very interesting thing happens. The left side field, which is coming from the right side of the retina on the right, and the right side of the retina on the left, are mixed and matched, and both sent back here. So we are visualizing the left visual field on the right occipital cortex. Likewise, the blue is mixed and matched and is on the right visual field is on the left visual cortex. Now, what that allows you to do is not only form images, but because these two eyes have slightly different visual fields. You think about it, with the nose sticking right here, there'd be a place where this eye is not going to be able to see past the nose, but where you have two eyes focused on a subject, you can compare the neural signals left and right. That's why we have a flat face with two eyes mounted pointing straight forward. That comparison gives us depth of field, the ability to judge distance. Think how many times that's important. So we were tree climbers, and with two hands we were grasping the branch. Is it important how far away that branch is? It is. That's distance perception. We are hunters. We're stalking up on a prey item, and we have to know how close do we get before we can shoot or before we can chase with a reasonable chance of success. That's distance perception. And that's what this crossover and comparison allows. In general, on the right side, we see here the auditory cortex on the temporal lobe, and a spatial visualization and analysis section, analysis by touch. And others on the right side, you can see the corpus callosum spanning the left to right side at the base of the fissure. And over here, functions like an interpretive center. This is language and math, auditory complex, and writing, the speech center that we mentioned before. In some cases, there is a clear one-to-one connection, like back here, visual cortex and occipital lobe is so well established. And problems with vision can often be related to the transmission of these nerve impulses from the retina back all the way through the optic nerve. So you can have a problem with vision anywhere along that path. The cranial nerves are going to close our discussion, and there are 12 of them. If you look at the names of them, the names are very suggestive of the function that they support. Because they're cranial nerves, they have a big impact on the functions in the neck and head. However, there is one nerve that's unusual, called the vagus nerve. Vagus means wanderer, and it leaves the skull region and goes clear through the neck, the thorax, the abdomen, with connections all the way down relating to autonomic function, and ending right in the beginning of the pelvic cavity. So here are 12 nerves, capital N and a Roman numeral is the way we talk about them, and all of them affect the head, neck, and face, skin muscles, except for cranial nerve number 10. Now, because of their location, they have a big impact, and there are specific cranial nerves that are donated to sight and the control of the eye, to hearing and the inner ear function of balance, to smell and taste. So the special senses are, the cranial nerves play a very important role in the special senses. I'm only going to review this kind of lightly. When you look at the inferior surface of the brain, here's the brain stem, here is the basis of the pons, and the midbrain disappearing into the cerebrum above. This is the base of the frontal lobe, and this tract running right here is an unusual a nerve. This is the olfactory bulb and olfactory tract cranial nerve number 1. It is the one that runs along the frontal bone at its base and disappears and connects directly into the brain and the olfactory center. It is not connected into the brain stem. The others, the optic nerve, here is your visual signal for the actual image. The oculomotor nerve, the muscle that moves the eye, and basically over here, the trochlear nerve, the trigeminal nerve, the abducens nerve are all involved in control of that eye region. Abducens, facial takes care of sensory on the skin and the face, and motor commands back to the muscles. Vestibulocochlear, we will learn about the inner ear and its parts, the vestibule and the cochlea. This has to do with control of hearing. Glossopharyngeal talks about glosso is tongue and pharyngeal is the pharynx at the back of the throat. The vagus nerve, here's the connection for that odd one that goes all the way down out of the skull, all the way to the pelvic cavity. Hypoglossal and accessory nerve are shown here in the bottom. To kind of illustrate only one of these, here is the nasal cavity cut away on a midline cut. The dental architecture that is shown here on the upper, this is the hard palate, this is the soft palate, and the frontal teeth, the lips, and the opening to the nasal cavity. This shows the pituitary gland. Notice that it's tucked into that sella turcica, the protective region which surrounds the pituitary. Here is the olfactory tract and olfactory bulb. Now the way this functions, processes, which just mean tubes filled with cytoplasm, come down through perforations. Those perforations are the holes in the cribriform plate of the ethmoid bone. They come down here and branch into dendrite-like roots, and there are sensitive cells on their surface. They are chemoreceptors. So as we breathe, here comes the air loaded with molecules. Some of them are scent molecules from lilac bushes. Let's say lilac bushes, because when they bloom, they generally have a very sweet smell. So when their air is drawn through here, those molecules hit the chemoreceptors here and cause an action potential. If that action potential fires the nerve, it's translated back here and interpreted as a specific scent, in this case the scent of lilac, by the olfactory cortex. You notice that was in the it was in the lateral sulcus on the side wall on the temporal lobe. Now, I want you to return to this diagram first. This is where you should begin and end. Be able to identify the 1, 2, 3, 4, 5, 6 lobes of the brain and assign a generalized statement of function to each one of them. That would allow you to get a start You must know some of the individual lobes, their anatomy and their function, before we can start to talk about how they're integrated in complex function. That's your introduction to the brain, and I'm going to end the recording